Hi, my name is Trevor Siegmiller, and I'm presenting on an adaptation of the influenza virus, a danger to public health. So, influenza, more commonly known as the flu, is one of the more common ailments that affect mankind. Most people will catch the flu several times within their lifetime, and for the average healthy person, it's usually only a mild inconvenience with aches, coughing, stuffed nose, maybe a fever. What most people don't realize is that there's actually quite a bit of destructive potential to the virus. In the year 2014 alone, 4,605 people died from influenza, whereas by contrast, in the early 1900s, there was actually millions killed in one of the worst pandemics in recent history. So, unlike other viral diseases such as measles, influenza vaccines are only relatively effective, um, and new vaccines must be generated every single year. This is due to the rapid evolution of influenza. The adaptability of the virus makes it difficult to combat, and treatments that were once highly effective become increasingly ineffective with each season. So, the question we're seeking to answer is, how does viral evolution affect our ability to combat influenza? So understanding the evolution of viruses is key to fighting it, as influenza is probably one of the better living examples of evolution at work. It adapts to overcome even the best efforts of modern medicine as it rapidly expands its phylogeny of species. In fact, we're still discovering new species of influenza, and we'll get into that a little bit later. But through this research, characteristics of influenza strains are observed, effects of viral adaptation are examined, that a more complete view of the role of evolution in influenza survival may be understood. So a few definitions for you. These will come up later, but just keep these in the back of your mind. Antigenic drift is the gradual drift in a genome of a virus caused by gradual mutation. Antigenic shift is a sudden shift in the antigenic character caused by a crossover with another subspecies. Glycoproteins are proteins that coat the surface of a viral envelope, and they carry certain groups that act as like identifiers, and it's what antibodies use to identify viruses. Quasi-species theory. So this theory, or the concept, is that the genetic diversity of a population is closely clustered around a single mutation or a single strain, with variations coming off of that. So we'll get into that later. So, history. So while some regions have flu seasons, as we do here in America, the global flu season is constant. Influenza outbreaks occur annually in many locations around the world, while pandemics occur on average of only a few times each century. So figure 1 here illustrates some of the more recent pandemics. We'll note the pandemic in the early 1900s and the death count that goes with that. It's an estimate of 40 to 50 million people. So it's important to understand the past pandemics. One, because it stresses the severity of the flu, and it shows, and simply the fact that a lot of these same exact strains that cause these pandemics are recurring. So, for example, the influenza pandemic of 1918 it's been described as the greatest medical holocaust in history. And what's interesting about it is that it's actually a very, very similar strain to what we had not just a few years ago with the swine flu. So in the early 20th century, very little was understood about virology, and even less was understood about the pathology of influenza. So with World War I still waging, it was an ideal opportunity for the spread of the H1N1 strain, as people were traveling all over the world in ships, as part of the war effort. You're living in confined barracks, hospitals, trenches, etc., and allowed the virus to spread like wildfire. So in only a few weeks, it spread from Europe to Asia, and by 1920, an estimated 40 to 50 million people had died in just a matter of a few weeks, which is pretty incredible, actually. And You see here, this is actually an image of one of the hospitals, and every single person in here is a flu patient. So the high death toll for sure leaves no room for doubt that influenza has the potential to be a significant health threat. In fact, the casualty count for that one epidemic exceeded that of World War I itself. In fact, it doubled it. So, as we see here, this is the outbreak pattern. And this is important because more common than pandemics are outbreaks. To really understand the evolutionary significance of this, we can describe it in ecological concepts. In ecology, an organism that adapts to fill a new unoccupied niche may experience rapid population growth. Influenza outbreaks function in pretty much the same manner. A new strain, one that is unique from previous strains, develops in one region of the world, such as in 1918 developed in Britain. A person or animal, in this case swine, who has developed immunity over the years to the many parent strains of that region, could unknowingly carry that virus into a new environment. So, for example, a farmer brings his cows or swine to the market. One that has not previously seen the virus or that particular strain, and thus people or animals around the carrier are infected as they inhale aerosol droplets or make physical contact with the carrier. Then the antibodies in their bodies won't recognize this strain, and the virus has the advantage and spreads very rapidly, and the process continues, and that's how you get it jumping all over the place. And this process continues every year with regional outbreaks, and this is the same process that led to the swine flu epidemic fairly recently. So to understand how that happens, we need to first examine the adaptation of the individual virus and its characteristics. So influenza is a negative sense single-strand RNA virus, as you can see here by this cross section right here. It comes from the family Orthomyxoviridae. It's an enveloped virus, which means it has a lipid membrane surrounding it with glycoproteins. That's what these are on the surface, and these act as signaling. Another cell will recognize it as being a cell's material and take it in, and it also, ideally, antibodies would recognize these and stop the virus, but that usually doesn't happen. So pleomorphic just means that it's able to alter its shape and size according to the environment. There are 4 primary species of influenza. Influenza A is perhaps the most common. It's the most common in bird species, but it is frequently passed to humans or other animals. It's the primary cause of pandemics, including the 1918 pandemic as well as the more recent swine flu epidemic. And it mutates at a faster rate than the other species. Type B is mostly found in animals. For the most part, you're not going to see it in humans. The reason why is because it has a much slower mutation rate, so the antibodies are able to keep up with it a little bit better. Type C rarely affects humans, usually only old people, so we're not really going to talk about it. Type D. So Type D is a very recent divergence. It was first discovered in 2011, and technically hasn't even been added to the official taxonomy of viruses. And there's also type E and F, which also have not been added to it, but they're so new that we're still figuring out whether they could be considered their own type or not. So adaptations. The danger of influenza doesn't come from those 4 main types, but it comes from the subspecies of those main types. Viruses experience much more rapid mutation and replication than most other life forms, as you see in Figure 3. RNA viruses in particular lack proofreading mechanisms such as we have in our own DNA to correct mutations. So as you see here, RNA viruses are the most rapidly mutating organisms out of here, whereas eukaryotes are down here. So significant difference there. It's through this that it really exemplifies the Red Queen hypothesis, that it's constantly adapting and thus is able to keep ahead of everything else, including our own cells over here. So this rapid mutation results in a high degree of variability and adaptation, leading to rapid evolution and a highly diverse progeny. This concept is described as the quasi-species theory. See here. The quasi-species concept refers to the genetic diversity of population clustered around a common genetic sequence. So in this case, in this example here, you have a single strain or single mutation type that develops other strains based off of that, and it gradually develops a sort of family tree of viral evolution until you have hundreds of strains based upon that one single mutation. And this is the quasi-species concept. So, and that's the result, is that you have the emergence of lots of new mutations and variations that have unique combinations and characteristics. This, combined with a high mutation rate, is why influenza is so adaptable and very difficult to combat. And it especially creates complications in vaccine development. And we'll talk about that in a little bit. Vaccines, here we go. Perfect. So, as you just saw, the mutation rate shows that viruses adapt much faster than us, which is why we require the aid of vaccines. So to really understand the mechanism of adaptation and, a basic understanding of vaccines is required. So for the last 70 years, we've been using an egg-based vaccine, and there have been other vaccines developed, but there's only 3 that are recognized by the FDA, and the last 2 have been developed within the last 4 years. So they really haven't made it into large-scale production yet. But the basic vaccine mechanism is the collection of a recent strain of influenza followed by incubation within an egg. The virus is then weakened and inserted into the human body, which allows the body to develop antibodies that recognize these specific receptors, or the glycoproteins, kind of like the identity on the viral envelope. And the body then identifies it as an antigen. So the problem with this is that it's effective against the strain that the vaccine was developed around. So that particular strain was in existence when it was developed, but that strain may have evolved by the time the vaccine is actually mass-produced, rendering the vaccine far less effective. In fact, in an analysis study conducted in 2011, they compared the efficacy of influenza vaccines in 5,707 different studies. And what they found is that vaccines have a highly varied level of effectiveness, particularly in the case of seasonal influenza, and that the effectiveness is either greatly reduced or completely diminished in some seasons. And they weren't exactly able to determine why that varied between seasons, but that's what they observed. So why does this happen? The mechanism behind it is called antigenic shift, as well as antigenic drift. And this is really what makes it so difficult to combat. So antigenic shift and drift, as described earlier, refer to changes in the antigenic character or the identity in terms of the antigen that is the influenza virus and the ability of the antibodies to recognize it. It's probably best understood if just drawn out. So let's say, for example, if we took a virus, an influenza virus, and we froze it, and this is a chart of time versus number of mutations. So if we froze it, that virus over time would not have any change in number of mutations because it's frozen. Whereas, say, if we had another strain that was isolated and just allowed to its own devices over time, you would see a pattern like this, where gradually you have an accumulation of mutations. And this is called antigenic drift, where over time the genetic makeup drifts, and your end result has a number of mutations from the beginning result. Antigenic shift is a little bit different, and it looks more like this. And this is caused by crossing over with another subspecies, and it causes very rapid mutation. You see this with influenza A, the pandemic variant of influenza, and this is why pandemics and outbreaks occur so quickly, is because of that mechanism. So this can result in a number of different combinations of glycoproteins and surface signal proteins on the viral envelope, meaning that human antibodies may not recognize it as the same antigen that the vaccine's targeted for. So this is normal attachment of a virus and cytosis of it into the cell. This isn't necessarily influenza, but same concept. And this is what would happen if an antibody recognizes it and attaches to it. So with antigenic shift and drift, the antibodies do not recognize the signals and glycoproteins on the surface of the envelope. So this, particularly in the case of vaccines, this results in varied efficacy, as different strains display varying characteristics that may have multiple characteristics from different parent types and strains. So in order to compete with the constant adaptations, researchers would have to predict the likely strain of the virus that will dominate the next flu season, which is why it is very difficult to do, and entirely possible that their prediction will be 100% wrong. Even if the vaccine is proven effective, unfortunately, it might not be long until the virus adapts to overcome the treatment. This is shown in the case of a drug called amantadine. In 2001, a drug, amantadine, started to be widely used in multiple countries. And as you see from this chart, this shows percentage of resistance over time. And initially, it was extremely effective in all these countries listed. And by 2006, just 6 years later, the virus is almost 100% resistant in almost all these nations. So within the course of only 6 years, through antigenic shift, it was able to overcome a highly effective drug. So that is the problem, essentially. So the future of antiviral treatment is somewhat complicated. There are many different treatments being developed in labs across the globe, with very different approaches to trying to combat the aggressive evolution of influenza. So typically what they're doing is they try targeting different stages of reproduction of the virus. We won't get into that specifically, but so instead of targeting antigenic recognition like our current vaccines do, they're looking at other things like targeting the RNA polymerase and other things that would completely halt the viral production. The problem is that while in theory this would work and this would be great, there just haven't been enough clinical trials to really determine whether these would be effective or practical on a large scale. So in conclusion, influenza is an excellent exemplar of the Red Queen principle. It's where the human race is caught in an evolutionary arms race of sorts with influenza virus. It adapts faster than almost any other life form in existence, and seems to always be one step ahead of even the best efforts of modern medicine, and able to quickly bounce back. As our efforts become more advanced, so too becomes the virus. So natural selection is giving the virus a distinct advantage because of its ability to mutate and adapt. And this, it's because of the crossovers, the quasi-species concept. These crossovers display a great amount of diversity, resulting in new adaptations, mixtures between subspecies, and characteristics with each generation. And this is why every year new strains arise from locations all over the world, causing outbreaks in other parts of the world as it spreads, which is why catastrophic pandemics occur with a relative frequency. However, ultimately, because of the rapid reproduction cycle of the virus, it's very difficult to predict upcoming adaptations and produce vaccines for those future adaptations and strains. Current treatments focus on our knowledge and our best guess of the next strain and of patterns, but current treatments consistently decrease in effectiveness with each coming year. This is shown in the case of amantadine, and was also shown in the analysis of those 5,000 different studies. Ultimately, RNA viruses, in particular influenza, have the evolutionary advantage, and that is due to the antigenic drift and shift mechanisms. Influenza may one day be one of the greatest health threats in the world if medicine cannot overcome its voracious evolutionary advantage and its adaptability. There are many new approaches being taken, but more clinical trials will need to be conducted in order to give us a better understanding of the viability of using inhibition of these different parts of replication. Further research could help researchers determine the effectiveness of different targets, and ultimately avoid future disasters such as a repeat of the 1918 influenza outbreak. Thank you very much. These are my references and sources.